Now, what we're going to do here today is we're going to have a look at a proteomic data set. And it's a dummy data set, and it's quite simple. We have 3 different cell types from an HIV study, and we're going to dive into the data and see how Omics Studio can help build hypotheses and also test them within the data. But good, let's have a look at the study preferences. We're just going to do it short, but of course a very important part is what kind of identifier do we want to look at. We're working with Proteomics, so we could look at the UniProt ID, but for most that wouldn't make too much sense unless you're a hardcore Proteomics guy. So let's just take gene names, that's easier to translate into what we're actually looking at. Then you have different thresholds and selection opportunities. Here we're just going to go with the default value filtration of 70%, and we're going to take all the samples and all the comparisons that we have. And then we also have some pre-selected preferences for enrichment analysis, and we're going to leave them on the default. And then I already selected what kind of database we're going to use for enrichment analysis. We're just going to look at Reactome, as we don't have all day today, even though I could easily use it. But perfect, let's save the study preferences, and then let's first look at the expression view, because this is where we can just get a quick idea of what's the quality of our data and how is the data behaving, and are there any outliers. And principal component analysis, as we're going to dive into first, is, for example, a good way to see if there's any potential outliers in your data. So we're just going to start it, going to choose our expression data set, and if you have multiple expression data sets uploaded, then you'll be able to actually select more of them so you can do your comparison in between as you dive into them. There's also a filter to my list. I'll not go into details with that just yet. We're going to look at it later. It's a very nice feature that we really enjoy using when we are using our software. Then you can choose different contrasts. Here we just have sample groups, but this is also where you could have gender, age, or clinical variables that you want to be able to visualize when you're looking at the variance within the data. Then we have the samples to include, and of course we've pre-selected all of them, and we're just going to move forward with these. Then you will also be able to select feature metadata, which is basically if you have some features that you would like to add to your IDs in the tables later on, but we don't have any for this data set. So we're just going to process, and then we're going to use the scatter plots, because that's typically the plots used for PCA analysis. And if we don't choose it, we just get the table, which gives less information in my view. But as you can see, we have the tables, we have it for the loading plot and the score plot, and we have our scatter plots. And you see, first we're looking at the score plot here. So here we have the different samples according to the variance from principal component 1 and principal component 2. And what is very nice with this cell study is that we can actually see some nice clustering of the different sample groups. But this would also be where you potentially could see an outlier if we had a sample that was very long away from everyone else. And you can switch between different contrasts. We only have one, so that makes it easy. But you can also choose different principal components, because you might think, I only see something in the small parts of the variance, which could be PC4. And when you click, it instantly transforms and you have that data represented. So very nice and interactive. And you can download the plot, of course. If we go to the loading plot where we see the proteins that was used for the plot, then you're also able to select the proteins. So you can use them for example, a list, which we're going to demonstrate later. So that was just a quick introduction to the PCA plot. For the total identification overview, it's very similar. Here we're going to look at if there's any unique identified proteins. Going to take our data set. We're going to choose our contrast. We have some value filtration criteria we can then use as thresholds. Here we're just going to work with valid values in at least one group of 70%, meaning that there needs to be an actual measurement of the protein within 70% of the samples within at least one group. And again, we have the metadata that we could add if we had any. And then we're just going to run the analysis, choose our plots, and it's already ready. So let's go and see. So here we have the total identification counts, we have the intersections. If we have, for example, some proteins that are unique to a specific, group, but we'll just quickly jump to the PCA plot because this is mirroring TMT data, and we have all the proteins identified in all of the sample groups. So that's a good validation for that type of data. Then we have summed abundance calculation, and I already made the calculation here because we're going to choose the same filters, but we're just going to have a look at the plots. And summed abundance calculation, here we'll just have a look at the bar chart, and there you can see what was the summed abundance of the proteins within the different sample groups. And here we can see that condition Y actually seemed to have higher intensity readings than the other two comparisons, which might be relevant later. You can also look at it in a box plot view, depending on your preferences, but also they will provide a little bit more information. For example, here we can see that the spread between the samples is actually quite low, which is good to see in a Proteomics dataset. Then we have the relative expression, which is basically where the data is, for example, log2 transformed when we are looking at Proteomics data. And here a very used plot is the heat map, which quickly give a good insight into how are the expression divided between the different samples. And here you also just be able to choose mean expression if you want to look at the groups more in focus. And we can, of course, switch around with how we're scaling it, how are we flipping the axis, if we want to hide the identifiers, and so forth. And we'll be able to choose different proteins, and then they will come to our current selections, and then we'll be able to add them to my list, which is a feature we're going to dive into very soon. But good, that just gave us a good quick insight into the expression data that we are working with. You can see that it's of decent quality and seems like there's something going on between the cells. Now let's look at the statistics data from the actual data set. Good. Looking at the statistic view, here we can of course look at the statistical data from our study, and we can choose which kind of comparisons we want to focus at. For this, we're just going to include all, so we have something to compare, and then it runs. And we can choose the plots that we want to have a look at, and you can see the data is already ready. So let's dive into it. You can see first, of course, the table from the statistical data that we loaded into Omics Studio. Then we can get dot plots and volcano plots. And if we just look at the volcano plots, because that's something we commonly address when we're looking at statistical data, then we can see the first comparison, X versus Y. And we can see there's actually a lot going on. There's a lot of proteins that are downregulated and also quite a few proteins that are upregulated. And comparing that with the other comparisons, then we can see that X versus Z, there was actually more upregulations, and diving down to Y versus Z, then we can see here it's also dominated by upregulations. Now it of course becomes interesting to see, is it the same proteins that are upregulated and vice versa? And this is where my list comes into play. So we're gonna dive into that one already now. So what we want to do is, of course, see that on the left side we have a bar for my list. So this is where they're gonna be stored. But when we're gonna create one, we can do it already using the statistic view. So we can take a select box And then, for example, take a look at the most downregulated proteins in the X versus Y conditions. When we select them, we can see them over here. And then there's no my lists right now, but if we click, we can create a my list and say, this is the downregulated in the X versus Z. And we can make a small description. I'll not go into that. The name says it all. And then we're creating it. And now we can see we can select it and we can add to it. So now these IDs have successfully been added to this list. And then you can see in the right part of the screen that we can actually select from my list. So if we click here, then everything disappears because we haven't selected a list yet. But if we do select the list we just created, then we now have a volcano plot that is just providing these proteins. And we can see, as we saw, X versus Y, everything is downregulated. But if we go to the X versus Z, then we can see that the proteins that were downregulated are actually upregulated for some of them in X versus Z. And if we look at Y versus Z, then it's actually the majority, well, it is the majority that are upregulated. So this could be very interesting depending on our different sample groups. And this is just one of the ways to use it. We can do the same with the dot plot. We can jump to them, and now you can see it's only showing 99 proteins out of the 18,000 data points that we have. But if we choose our list and just plot them on, then we can set it down to just looking at those proteins. We can see it's still too many for actually getting a good insight into the different IDs, but here is a good feature. We can zoom, but we can also adjust the height of our plot. So for example, we could set it to 1600 pixels instead, and now we actually can see the different proteins and see what was the fold change in the different comparisons, which is localized at the bottom of the plot. And we can see there's also some size difference to the dots, which is corresponding to the log 10 negative p-value. So this provides us with some good insights and ability to compare between our different comparisons within the statistical data. So this allowed us to make a list. Let's, for the fun of it, just add one more list so we can use that later on. We also want, I'll say, the upregulated in the X versus Z. And then, of course, we need to select these. And it's fast and easy, and we've chosen the right list and add to it. Now we can use it later on. But let's dive into the enrichment analysis. Here we have two opportunities. We can do an ORA representation analysis, and we can do a gene set enrichment analysis. We are going to start with the gene set enrichment analysis and also start the ORA representation analysis, because that takes a little bit longer. So we might as well have that running while we're looking at the gene set enrichment analysis results. But first, looking at the GSEA, here we can choose different thresholds. And for gene set enrichment analysis, it's of course best not to choose any thresholds, because it's going to be represented in the ranking from the enrichment analysis anyway. So let's continue and choose the database. You can see from a pre-select, Reactome have already been selected, and that's also the one we want to go with. So we can just hit process, and while that one is going, we can start our ORA representation analysis. That one already jumped into the selection of our database. We'll just quickly go back so you can see the filter we're applying. Here we are looking at everything with a p-value below 0.05, because we want to be exploratory. We could also add more strict criteria, but for this demonstration we won't. But we'll choose the reaction pathway here as well and hit process, and then let's go to the gene set enrichment analysis, which we can see is now done. We're going to look at bar chart and dot plot from the results, and again we have the table with all the different values from the calculation. We have the opportunity to find different filters and put in different criteria for which pathways we're going to look at, or we can just browse through all of them, but that's going to take a while. So let's jump to the bar chart, and as before we're only showing 100 data points, as there sometimes are too many for us to be able to render at a decent speed, and also for you to be able to actually see. Let's look at top 15, and as you see, instant filtration, and we can see which pathways are there. And more importantly, as we had our different conditions, we can actually see it from all different comparisons, so we can compare them. This can also be done a little bit faster by just going into the cross-group comparison. You can see right now we just did the enrichment, which will give the individual enrichment results. But if we go to cross-group comparison, then we get it from all. And when we choose top 15, then we get a quick insight into how these pathways are behaving in the different comparisons. So we can see X versus Y seem to have quite a unique pattern compared to the others. And it's very clearly presented here. And you can click on the different pathways, and then you can see it actually goes into your selection box. You can exclude it if you want, but you can also see the related identifiers. So these are all the proteins that are related to that pathway. And here you'll actually also be able to take a list and create it related to the pathway, which could be the one in focus, and use that later on. Just remember to push add to my list. We're going to use that later on. Let's quickly look at the dot plot. We have seen what it is before, but it's always nice to see it related to the results that we are looking at. And again, we can do as before, compare between, and we can do the cross-group comparison and compare them more directly in a one plot. But good, very short insight to it. And next up, we're gonna actually use some of the lists that we have created, and we're gonna use them in database search. Because here you're gonna get the opportunity to extract data from a lot of different databases to actually get more insights into the proteins that you have selected, or metabolized, or transcripts for that sake. So you can set a scope, what you want to be able to actually see in the different tabs. So here we have molecular identity, we want to see that. We also want structural and features and function interactions. And in theory, everything is interesting, but let's just keep it at these four and then say we want data from Reactome, and we want it from clinicaltrials.gov and from the Human Protein Atlas. And yeah, let's take UniProt just because we can. And then you see, when you click in, nothing is here, but that's because we just need to choose our lists, and then instantly we get some information. So, and we can go through the different tabs, or we can just look at everything. For example, we can see that from these proteins, there's only information from one protein from the Reactome. If we then go to UniProt database, there should be more, as is from UniProt identifiers. And here we can see much more data. And we can click to our focus point, which could be functions and interactions.
So for the UniProt knowledge base, we, for example, can see what other proteins does the individual proteins react with, and you can see information from gene ontology, molecular functions, and so forth. Something that's also very interesting, and information we'll get from the Human Protein Atlas, is where is our protein, localized? Where have it been found in other data sets? So here we can, for example, see some seems to be very represented in the breast tissue, and some in the cerebellum and cerebral cortex, and so forth. So here you can get an insight to if there's some specific expression patterns to different proteins. And we just selected one of our lists. We can, of course, quickly switch between them so we can view the data that is relevant. And now let's go back to ORA, because that should be ready, and it is. And again, we have our matrix with our data, we have our bar charts, same ability as we have with the gene set enrichment analysis, and just provides us a nice, easy access to the data. And everything is responsive, and we can pick up data points that could be relevant later on. So that was a quick demonstration of what you can do, how you can build hypotheses in Omics Studio, and how you can find information related to the proteins of interest, and also how can you do some light validation of them. For example, with the protein pathway of interest, we could also have just viewed that in our statistical information to see if it actually was relevant. So good. This was one of the many workflows that you can do in Omics Studio. And I'm sure you can think of several more. I can at least as well. But I don't want to drag you through a 4-hour session of all my different ideas of how to explore the data.